Welcome everybody to the MTB's fourth educational webinar, and the talk for today is Scaling Precision Oncology in or Out of Reach, and we're absolutely delighted to have our guest speaker, Dr. Benedikt Westphalen.
Yeah.
So,
Ah.
sorry,
Hi,
did I go
everyone.
through
I
to
just wanted to say hi.
you? There should be an opportunity to have some questions and answers after the talk, and you can either interact by asking questions in the chat box, or you can raise your hand and ask directly. I will be allowing everybody to be able to come in and speak, turn their camera on at the end of the talk. And to let you all know, the meeting is being recorded and the meeting is awarded 1 RCPI CPD credit. So I'll hand over to you now, Dervla.
Thank you very much. And we're really absolutely thrilled to have Dr. Benedikt Westphalen to give us a talk today on scaling precision oncology in or out of reach. Dr. Westphalen studied medicine and molecular biology in Hamburg, Philadelphia, New York, and Bern. And after graduating medical school, he started training in internal medicine at the University of Hamburg in 2008. In 2010, he joined the laboratory of Timothy Wang at Columbia University in New York City as a postdoctoral fellow, and his studies were focused on cellular plasticity and the origins of GI malignancies. After his postdoctoral training in 2013, he joined the Department of Hematology and Oncology at the University of Munich, Germany, and here his focus has been on clinical and translational research in gastrointestinal cancers with a focus on pancreatic cancer and rare cancers. Since 2017, he heads the Precision Oncology Programme at the Comprehensive Cancer Center at University of Munich, Germany. And since 2021, he represents the Comprehensive Cancer Center Munich within the European Reference Network for Rare Cancers, EURECAN. He has received research support from the German Research Foundation, the Universities of Hamburg and Munich, the German Cancer Consortium, the German Cancer Aid and Young Investigators Award from the AACR and the GRG AGA. He serves as PI and co-investigator on numerous clinical trials and has authored and co-authored more than 100 research papers. Dr. Westphalen participated in the 2018 ESMO Leaders Generation Programme, and he joined the ESMO Translational Research and Precision Medicine Working Group, as well as the ESMO GI Faculty Group in 2019. From January 2024 onwards, Dr. Westphalen chairs ESMO's Precision Medicine Working Group, and he currently serves as an officer for the German Cancer Aid and the AIO. In 2022, Dr. Westphalen has elected to join the EU Commission Mission Expert Group, Mission Board for Cancer. In 2024, he was nominated to join as a member of the Forum Zukunftsstrategie. Sorry, I thought I'd do a better job of that one, of the German Federal Ministry of Education and Research. And we are more than delighted to have him here today to give us a talk as part of the Irish Molecular Tumour Board. Thank you and very welcome, Ben.
Now I'm blushing. Thank you so much for the kind introduction. Let me pull up my slides. You should see them right now, and they should be moving. So it's an absolute pleasure to discuss precision oncology with you guys today. And maybe as a little bit of an introduction, I've become a little bit more political over the last couple of years. Being in the field of precision oncology for almost 10 years now, I've seen firsthand what is working, what we've promised, what is not working, and what we need to do better. And this is today's topic. So if we want to bring precision oncology to our patients, how do we do that? Is it a dream or a reality? Those are my conflicts of interest, none of which have influenced today's talk. I'll show you one slide that was prepared together with Roche, but the input was done by us. So what are we facing today? Cancer is a global problem. You all know that, being in oncology. We say 10 million people die of cancer each year. And in Europe, this is almost a fifth of that. Almost 2 million Europeans die of cancer each year. Why am I starting with these numbers? As of today, precision oncology is mostly practiced in later stage patients. So those are the patients that are succumbing to their disease And this is one of the questions that I might want to discuss with you later. Is this really the right thing to do when we talk about delivering precision oncology, that we always concentrate on the very last line when we're looking for a silver bullet? And this is a slide that I often start with in these talks in response to the Profiler 2 clinical trial by Olivier Trédon and colleagues in France. Ian Tennock wrote a devastating opinion piece in Annals of Oncology, where he said that precision oncology, the way we do it right now, is failing. Why? Looking at data from more than 10,000 patients in clinical trials that did precision oncology in the last line setting, trials such as Shiva, Profiler, or Moscato, you have a common theme. If you test 1,000 patients with comprehensive genomic profiling, roughly 40% of them will have a targetable alteration. That in itself is not bad. But the problem really is that only a small fraction of the patients for which we have a treatment option will in the end then be being treated. And this is owing to the old data that Ian was looking at. Only a small fraction is actually responding. And this is why Vinay Prasad started a precision oncology Twitter shitstorm a couple of years ago, when he, and then following up with a Nature Medicine perspective, claimed that precision oncology on a whole scale is not working and will maybe never work. So is the talk over by now? Luckily not. Over the last couple of years, we've seen numerous examples of molecularly guided treatment options coming into the market. This is data that we've assembled together with Lotte Steuten, Joaquin Mateo, and Emil Wust for a manuscript in Nature Medicine, in which we concentrated on the fact, how are we actually delivering precision oncology to our patients? And as you can see, this list is constantly growing. So I want to build my talk as of today, after this short introduction, around a couple of themes. We really need to rethink cancer care. And the first questions that you have to ask yourself if you're working in the field of precision oncology is A, do I have access to quality ensured testing? Secondly, if I have access to testing and I get the results back, am I able to act accordingly? And then last but not least, and this is critical, do my patients have access to treatment? And this is really the first question. Now in precision oncology, there is an intimate relationship between diagnostics and treatment. And your molecular pathologist is really the go-to person. You, as the treating oncologist, have to be able to talk to your molecular pathologist, understand what the testing can do and cannot do. Only then is this intimate relationship bearing fruits in terms of actually delivering treatment to your patients. And this is why I welcome a European Master's in Molecular Pathology that I have the pleasure to be on the teaching faculty with, by Marius Illier from University of Côte d'Azur in Lille, France. They have built a European Master's program for young and middle-aged pathologists going into molecular pathology, especially with a focus then to be part of a multidisciplinary team to deliver care to our patients. Why is it important that we as oncologists have the expertise to act on the results? This is legendary data by now by my dear colleague Stacey Gray, now at City of Hope, back in the days when she was still at Mass General. And what Stacey and colleagues did is, at a stellar institution, she went around and spoke to her colleagues in oncology, asking them about how comfortable they felt, she called this genomic confidence, to implement comprehensive genomic profiling, they called it multiplex genomic testing at the time, into patient management, and especially also to explain it to their patients. And you could say, well, in 2016, 50% of colleagues were able to integrate multiplex tumor genomic testing into clinical care. I myself trained in internal medicine, and I feel somewhat confident in treating high blood pressure or diabetes, but I would never be wanted to treat it by myself. So looking at the data over here, 30% of the colleagues were not very confident or not confident at all. And this is really important. If you get these testing results back, you have to be able to integrate them properly to see where there's therapeutic potential and where not. And this holds true. So now, more than 10 years later, no, not exactly 10 years, but 8 years later, Stacy and colleagues again went around and spoke to colleagues across the US, now almost 1,300 colleagues being interviewed. And you can see that there are still very significant number of colleagues that are moderately confident or not or little confident. So knowledge about the potential of comprehensive genomic profiling, what your testing can do, how to implement it into care is really important when you're building precision oncology programs. And when we're talking about scaling precision oncology on a regional or national or even international scale, then building expertise is critical. Why is it important? This is data from the LungMap study. So the LungMap study investigated the impact of comprehensive genomic profiling in the management of non-small cell lung cancer. So a no-brainer. And overall, only a fraction of patients, regardless of their household income, race, whatever, were able to understand why they were on LungMap. They thought that their doctors wanted to find out if they really had lung cancer. So if you're not able as a clinician to explain properly what your profiling molecular diagnostics is for, your patients might even consent onto a clinical trial without actually knowing why they're on. And there was a nice editorial in Nature quite recently that is asking or advocating for a genomic savvy healthcare workforce, so that we need teams of healthcare workers that are able to optimize delivery of patient care. And the last question, and I briefly alluded to this manuscript then, is, do my patients have access to both testing but also therapeutics? And this cannot be focused on a few academic centers in privileged countries. If we want to deliver precision oncology to patients with cancer, wide and equal access is key. And I know that this is a critical issue, but there are ways to ensure access to testing across the globe. And one of them, Emil Wust, Hans Gelderblom and team cannot be congratulated more, is what they have done in the Netherlands, so-called drug rediscovery protocols. So what Emil and Hans and their team did is they had the infrastructure for comprehensive genomic profiling for a majority of cancer patients in the Netherlands. And they had molecular tumor boards, and they said, well, now we need to treat patients. And together with pharmaceutical companies on one hand, but then also payers, insurance companies, regulators, they built a nationwide clinical trial program where drugs that were approved in one indication, molecularly guided drugs, could be used in the setting of the drug clinical trial. And the pharmaceutical companies would give out the drug for 4 months. If after 4 months the patients benefited from treatment, then the insurance companies would take over and patients could be treated until progress. And this goes into both ways. In cohorts where you would find out it works, there was a road and there is a road to conditional approval. And one example being nivolumab for MSI cancers before Pembro got broad approval in the EU. And then there's also cohorts where you say it just doesn't work and patients will never get access to these drugs again because in a clinical trial setting, they failed on a national scale. So after answering these questions to yourself or within a healthcare system, the next questions that arise is, what information can I get from testing? And there are three questions that I want to circle around. First of all, do I test according to standard of care? Secondly, does testing inform my management? And last but not least, do I screen for clinical trials? Starting with, do I test according to standard of care? We at ESMO have recently published the second iteration of our recommendations for the use of next generation sequencing for patients with advanced cancer. And you can read the indications that we have included yourself. What I would want to stress here is that this is actually very solid from a methodical point of view. We looked at every given cancer, how many therapeutically and clinically relevant biomarkers are in a given indication, so that if you test all of them at once, your patients benefit from the chance of an in-label medication. And not surprisingly, one of the first examples being non-small cell lung cancer. I believe there are 8 or 9 targeted agents approved in the first line setting. But then there are other cancers, such as colorectal cancer, where if you treat according to international guidelines, if you test according to international guidelines, MSI, BRAF, ALRAS, HER2, all of these biomarkers can be assessed by comprehensive genomic profiling. And if you do this within a one-stop shop, you are most probably more tissue sparing, but also cost effective. And I can show you that for a growing number of agents now, also tumor agnostic approvals have been seen in Europe and in the US. So you have drugs that are approved that are mostly or linked to a genomic biomarker that can only be assessed, for example, NTRK or RET fusions in a comprehensive manner by advanced molecular diagnostics. So with at least 2, and we'll most probably see a third agent entering the market in the next couple of months, you have a growing number of molecularly guided treatment options that are sort of linked to comprehensive genomic profiling. So the standard of care here is really changing because of the growing number of tumor agnostic approvals. And this is the slide that we built together with Roche for a symposium at ESMO 2023. And what I want to show you here is really this tsunami of molecularly guided drugs that are about to enter the market. What did we do here? So you can, as a rule of thumb, roughly 30% of drugs that make it into a phase 2 or phase 3 clinical trial will in the end up being approved. We were more defensive and set the bar at 20% and looked at all clinical trials that were recruiting based on a genomic biomarker, and then estimated if only 1 out of 5 is going to end up with a label, we're going to see this huge increase of molecularly guided treatment options in the coming years. So the second question then being, does testing inform my management? And here I want to go back to the example that I briefly alluded to when talking about Fernanda's paper in the first place. So if you are testing according to international guidelines, in colorectal cancer, you need to test for KRAS and RAS mutations to rule out these alterations to be able to treat with anti-EGFR monoclonal antibodies. Then there is the question of BRAF testing. Then there is MSI-DMMR with your PD-1 checkpoint inhibitors around. And then there is growing evidence for G12C inhibitors, now with 2 compounds in the market being ranked SCAT1A. And then, of course, there are less frequent biomarkers that we're going to see in the future, even more importantly. And then staying a little bit with the example of colorectal cancer, but also pancreatic cancer, there are roughly 3.5 million patients diagnosed each year with RAS mutant cancers. And since almost every pharmaceutical company around is now testing RAS inhibitors, being isoform specific or pan-RAS inhibitors, it's going to be critical to know which patients are actually RAS mutant, not only in the management of colorectal cancer. And just to showcase you one example from last year's ESMO, RMC 6236, a RAS selective multi-on inhibitor, so RAS mutant specific, showing you data from the pancreatic cancer cohort. So this is heavily pretreated patients on a monotherapeutic regimen, with a huge number of patients being stabilized, and you see there are even partial responses. A year ago, these 46 patients were only on treatment for a very, very short period of time. And I can tell you that RMC, and I don't work for them, I don't receive any sort of royalties or so, but they have announced that they're going to show updated data, and a lot of the patients that were reported at ESMO 2023, so I hear, are still on treatment. So the short duration of response that you see here is owed so that to the fact that this trial was reported quite early. Last but not least, what information can I get from testing? Do I screen for clinical trials? This is data from the MSKCC Impact Program, where all patients that enter the door of this prestigious institution undergo comprehensive genomic profiling in a fully automated fashion. So sample is collected, everything is done by robots, so to say. And then if there is a clinical trial at site, both the molecular pathologist, the treating physician, and the PI of the clinical trial will be notified by the automated system that there might be a match in terms of a molecular biomarker. And by doing this, the colleagues of the MSKCC are able to enroll more than 10% of their patients into molecularly guided clinical trials. And it is no surprise that a lot of the seminal clinical trials and approval trials for rare and very rare alterations, such as NTRK, were driven by this approach. A lot of cancer centers, including my own, would be happy if they would have enrollment rates of 10 or more % in the whole cancer population. So these 11% in later stage patients are truly remarkable. So if you have a structured access to testing and a good clinical trial program, these two things really go hand in hand. So far, so good. So I hope I was able to convince you that the landscape of molecular insights and molecularly guided therapeutics is rapidly evolving, and that access to quality ensured testing, expertise, and treatment are pivotal. Comprehensive tumor profiling can inform your standard of care, remember colorectal cancer. So port screening for clinical trials and provide caregivers with additional clinical information. And if we're thinking about scaling precision oncology, profiling is not a nice to have hobby of a motivated molecular pathologist. And then there are some doctors that believe precision oncology is really cool. So this has to go hand in hand. And this is a question of integrated healthcare solutions in the end, where testing and treatment need to be linked together. So I believe we're standing at the crossroads, because the question was, how can we scale in the end? And if I was building a precision oncology program again, it would be around certain questions and building sustainable structures. So we were lucky enough to be allowed to write a consensus paper now 4 years ago, published in the European Journal of Cancer, for what we believe would be important to build a nationwide precision oncology structure. And as you can see on the right side, there is the question, who should undergo testing? And this is going to be super important in the future, because all healthcare systems are under immense pressure, and working smartly with the resources that we have will be more important. We've touched upon education. It is important in terms of harmonization that we speak the same language, that we define what a molecular tumor board is. And then I cannot stress this enough. Think about access to treatment in the end. It is important that we collect the data that we generate, and then finances is critical as well. There are numerous examples of precision oncology programs across the globe. And what I've learned over the last couple of years, and that's why I'm so grateful to have this role within ESMO as well, is that the precision oncology community worldwide is extremely friendly and cooperative. I had the immense pleasure to work with the colleagues from India to implement the Indian molecular tumor board called Impact India. I sometimes sit in and learn incredibly from these colleagues. And so if you're thinking about building structures, I'd say reach out to colleagues. I've never been turned away by anyone in this field. Maybe it's because it's a small and tightly knit community and we all like each other's mostly. But I believe in the power of teams. And so if I was building again, I'd stress the team building effort even more. And together with Raffaella Casolini and a lot of other colleagues across the globe, we are part of a Lancet Oncology Commission, really looking at the challenges and concrete solutions, how we can, in the end, accelerate precision oncology in healthcare and research across the globe. So, coming back to the question, how can we scale? First of all, what could help? Technology, for example. So this is taken from the ESMO guidelines on liquid biopsies. You are well aware that liquid biopsies can be used for various questions in the setting of the management of cancer. What I believe they could play a role with is if you have a decentralized healthcare system, such as Germany, and you build structures of reimbursement and quality control, your patients and tumor samples from 10 years ago do not travel throughout your country, but their blood travels, and the comprehensive genomic profiling is done at centralized structures. And there's beautiful data that I have not included today with novel technologies and readouts, such as the so-called tumor fraction. You can be quite sure if your liquid biopsy is informative. Back in the days, if we wouldn't find any alterations, we were not sure if our testing actually worked or the tumor was shedding. Now with tumor fraction, there's data from Cristiano Rolfo If you have a tumor that is actually shedding and the liquid biopsy is negative, it's a true negative. And if you then test the tumor sample, you won't find anything else. So here, I believe, in terms of scaling, if you build sustainable structures, liquid biopsies could be helpful. And this is the same is true for the clinical trial landscape. It is not imperative that everything is done at site. So there's the concept of decentralized clinical trials. Spain, for example, is running them where some of the intervention can be done close to home, and only certain things are then done at the actual clinical trial site. I used to say that this is clearly nothing for CAR T cells, for example, but since the Americans are able to do CAR T cells in the outpatient setting, I will have to look for other examples here. But the point being, really, if we think about smart structures, then access to treatment could be broadened because patients are not confined to just the one cancer center in the capital in the end. And then I believe that we could use artificial intelligence, and this is now already legendary data by my good friend and colleague Jakob Carter, who was able to show, by the way, fully self-trained. He was then a young trainee in gastroenterology with Tom Lüde and team, self-trained AI mastermind. Jakob and team were able to show that you can use simple H&E slides to predict microsatellite instability. And here we're now seeing a lot of development where you actually go one step ahead and you say, I don't need to do the broad comprehensive genomic profiling to screen for rare targets. I use artificial intelligence enhanced pathology to do the pre-screening and then do confirmatory tests in those that come out positive in these pre-screening situations. Now you say, is that really a way to scale precision oncology? I'd say yes. Why? Going back to the data by Miriam Shalabi and colleagues who were able to show that there is a role for neoadjuvant immunotherapy in clearly operable colorectal cancer. We're thinking about scaling biomarker testing in a cohort that we would normally not screen. A lot of the patients in Germany go from the gastroenterologist who do the endoscopy straight to the OR without ever seeing an MSI test. So don't get me wrong, I'm not advocating here for neoadjuvant nivo-ipi for all patients. I'm just giving an example where screening of large cohorts and then changes in the management of patients could be supported by artificial intelligence in situations where you would not normally order biomarker testing. And I believe if we are thinking about such impactful data as Miriam has shown or Andrea Čaček's group has shown with rectal cancer, it's a question of democracy that you shouldn't have your patients being tested by chance or screened by chance. This is stuff that needs to go to the population. What will happen in the future? I believe there is great potential learning from precision oncology and taking this into multiple cancer early detection. More than 140,000 patients being enrolled in the NHS Gallery trial in the end. So I believe we're not going to stop at late stage patients. We're going to see more patients in earlier lines of treatment. And I know for a fact that a lot of pharmaceutical companies are already preparing their later stage compounds and assets to be used in earlier lines to do neoadjuvant and adjuvant treatments with targeted agents. So this is one thing that looking into the next 5 or 10 years, if we're thinking structures now, at least we have to keep in mind. And then maybe even more into the future, brilliant data by Charlie Swanton and team presented at last year's ESMO, where Charlie was really able to show that a subset of patients with EGFR mutant lung cancer due to air pollution carried a specific set of inflammatory phenotype that could be reversed with anti-inflammatory agents targeting IL-1 beta. Why am I looking that far into the future? We're now seeing technologies and knowledge that we generated in the setting of cancer research being applied in maybe precision prevention as of now. So this is just a look into the future, but if we consider this now out in the public domain, we have to think about structures around this as well. And then last but not least, I'm convinced, because I'm seeing the first clinical applications here, that we're going to use artificial intelligence to better understand patient populations that might or might not benefit from a given therapeutic intervention. This is also personalized or precision oncology, where you say, this is a high-risk patient. He or she will not benefit from standard of care first-line chemotherapy. Let's see if we have a clinical trial at hand. And since these AI models are so incredibly powerful and can integrate a lot of data from a simple H&E slide in the end, I believe they will help us scaling in the end. So summing up, and I'm really looking forward to the discussions after this, I believe precision oncology as a concept has the potential to fundamentally change our approach and perception to public health, because it is this connection between diagnostics and treatment that you cannot divide away from each other that allows for personalized treatment. We're now seeing the first questions of personalized early detection coming up, and then also personalized prevention. And if we believe that precision oncology is a concept that is going to stay, and I'm convinced of that, we have to realize that this has socioeconomic, in the end, targeted treatments and diagnostics cost money, and ethical implications. Who should be tested? Who should go onto targeted treatments? Is this something that we as a society can deliver upon in the future? I've alluded to the concept of integrated healthcare solutions, where you do not say the pathologist is doing the testing and then someone in the Department of Oncology or wherever the point of care is doing the treatment, and those two things are not linked. Here I see great potential for pay for performance models, both on the testing and on the treatment side. It underscores, and this is critical, the need for true collaboration and multi-stakeholder transsectoral approaches. So coming back to the question, scaling precision oncology in or out of reach. I believe if we continue doing it the way we do right now, silos, so forth and on, it's going to be out of reach. We alone, we get to the base camp of Mount Everest, but only if we work in teams together, we will make it to the top. And this is why I'm always ending my talks with the call to action. There is a need for a leap. We all need to work together to bring all of these components, all of our common knowledge in terms of shared intelligence together to achieve the best for our patients. And with this, again, I'm very grateful for the invitation. I hope you found this okay-ish, and I'm really looking forward to the discussion.
That's fantastic. Thank you, Ben. That was a brilliant run through and covered so many different topics of molecular tumor boards and precision oncology. So I'm just going to open the floor to some questions But while I'm waiting for that, I had a couple of my own, if that's okay. So I suppose just going back to Vinay Prasad, have we just got better at pretending that we're still living the illusion? Are we just testing much more, identifying more, but actually we haven't moved very far from Ian Tannock's 3% actual benefit? Or is that, that was 2016, that was 2017, 2024 and onwards will be a very different landscape?
So I believe we've gotten better. We have a huge public relations problem, so to say. I mean, oncologists are always very eager to jump onto the this is new and fancy train. And I believe what we've done with comprehensive genomic profiling, we have advocated like hell. We need to do it, we need to do it, we need to do it. And the pharmaceutical industry has banged this drum as well. And now we're standing, and this is what Ian was alluding to, in front of a mountain of shitty data that we have to defend. It doesn't work if you're looking for the silver bullet, where the question is hospice care or a silver bullet. You have to know which patients benefit from testing, and you have to know which patients do not benefit from testing. It breaks my heart to say it, and I don't know if Grania is still on the call, but in a 78-year-old pancreatic cancer patient, the chances of you finding a RAS wild-type pancreatic cancer case are so incredibly low that I wouldn't say this is a good poster child for precision oncology. So we've gotten better in understanding who needs to be tested, and then we've gotten so much more better in knowing which drugs to use. So the simple example being PI3K mutations or AKT mutations. Those were addressed in all of these clinical trials with mTOR inhibitors. You can throw an mTOR inhibitor at everyone, and most of the time they do not work that well outside of breast cancer, for example, but or NETs. Now we have AKT inhibitors, and if there was an alteration and you have access to this drug, your chances of actually delivering to your patients are far better. So that would be the answer to this question, hopefully.
Yeah, no, good. So the future hopefully is just we're getting better, and we're getting there. I wanted to talk about finance and toxicity, and Brian Bird, who's one of our oncologists, who's Billy the oncologist, not quite Billy, Brian the oncologist in Cork, in one of the hospitals here with me. So I suppose the explosion in precision oncology has gone hand in hand with biopharma drug development, and you can see that from the future planning. We've got more and more inhibitors coming, more and more targetable. Or more and more compounds that can target. But of course, biopharma ultimately, and I hate to say it, but it's true, have at the heart, they're for profit, and they want to make money because they want to do more R&D, but also because, they want to make some money. So the financial toxicity of drugs is enormous. And I suppose, how are we going to address that in the future? Are there kind of global partnerships that we'll be able to think of, especially not just for low and middle income countries, but there's also countries in what would be considered high income countries that are low resource. So even, for example, even Ireland, we are very slow to access any of these targetable treatments. So Germany gets them years before we might. And that inequity for patients always sits a bit kind of poorly with me. So how are we going to address that from your opinion? But also just to flag, because Brian has suggested obviously something, should drug companies be held accountable for patient outcomes, so that if a patient derives a benefit, that then you should pay the money should be paid to the drug company. But if actually a patient doesn't derive benefit, then that would be, I suppose, pro bono. And then obviously should a testing company only be paid if there's a therapeutic finding? So they're kind of two potential, I suppose, ideals. But I wondered about your opinion on those and your own kind of ideas.
Yeah. So this is a political problem that we're facing here. I mean, drug development has gotten so incredibly complicated, regulated, and therefore expensive that a pharmaceutical company that is still in the R&D field, in the end, in order to be held accountable or, to their stakeholders, need to have a financial end game in the end. Academic drug development is essentially that, doesn't work anymore. So we're stuck with pharmaceutical companies as the major driver of therapeutic innovation in the field of medicine, I would say. This is maybe just a side note, but this is why we're not seeing new antibiotics or antivirals. If you're only on a drug for 2 weeks, at a time, that's not a huge market for a pharmaceutical company. So with regards to then risk sharing, pay for performance models, I find this extremely attractive because, and this is what I mean with integrated healthcare solutions. So I would echo Brian here. You can easily, on the testing side, imagine, let's say it's 3,000 euros a test. You get 1,000 euros if your test is technically successful. So you say you better be good in terms of quality. So 1,000 euros is roughly what you put in terms of sequencing costs, so forth and so on. You get 2,000 euros if the test is informative. That can be either informative in terms of someone being RAS mutant and you do not give EGFR inhibitors. And then the third portion of money is given out if the patient actually benefits from what you found in your informative test. And this, of course, you could carry on into pay for performance models with high pricing drugs, where you say, I believe 6 months is long, especially if we're moving into later lines. But somewhere in the range of 3 to 6 months, maybe. And we in Germany have at least one example, namely CAR T cells. So these drugs, it must be the same with you guys, are extremely expensive, and the insurance companies only pay for the full price of the product if patients are benefiting at a given staging interval. So yes, these are models where where I believe one could work with regulators and payers and pharmaceutical companies to bring these drugs into the market. Absolutely.
Yeah, I suppose my concern, Ben, about something like that is if pharma aren't being paid for, 4 patients and then are get paid for their success, that the cost they've lost from those other patients will fall onto, it will ramp up the cost for the individual patients. But these are things that we need to work out. But I totally agree. I mean, I think this is one of the biggest issues globally that we have at the moment is how do we afford cancer diagnostics and cancer care now and moving forward into the future, which, as you can see, is only going up.
Maybe just one point to that. So, I mean, I cannot stress too much what Emil and his team in the Netherlands have done. So this multi-stakeholder approach, going to the insurance companies, going to the state regulators, a strong academic backing, all of that as a public-private partnership. And stuff like this has also happened in other countries, Norway, Sweden, so forth and so on. It might be complicated in other healthcare systems. It's most probably not working in Germany due to regulations and law. But this is an example where you can learn a lot from how they actually set it up, and that biopharma partners then followed suit.
Okay, great. So Siobhan has a question, if that's okay. So Siobhan, go ahead.
Thank you very much. Fascinating talk. I'm an advanced cancer patient currently, but actually previously worked in gene therapy. And gene therapy is another example where you pay for efficacy. And the first gene therapy in the world was licensed in 2017. So actually we now have, good data. If you look at gene therapy, although it was a rare disease, so all of those things, it took a long time to get up and running, but actually Novartis, who now look after the gene therapy I worked on in Europe, have now figured out solutions in some of the jurisdictions. So there are precedents in gene therapy as well, just to add that. The second question I wanted to ask was, from a patient perspective in advanced cancer communities, there's a lot of talk about precision medicine. Naturally, a lot of patients are going out and getting next generation sequence themselves, independently of their hospitals, their centers. And I was just wondering if you talk about the problems that may pose, or if you're in favor of it while we're in this interim situation.
Yeah, so my feeling is, and this is on all of us. So, I mean, you've seen that I've worked with, advised a lot of pharmaceutical companies, and I see that a lot of them, when they do patient advocacy work, is that they put you up front to ask for more and more testing being done. And it almost feels like that they are putting by proxy pressure then on the caregivers. You have to ask your doctor, if you're not tested, then he or she is not doing you right. I believe that we have, and I myself, maybe 4 or 5 years ago, would have said, the more we test, the better. But the more we tested, the more we learned where it just doesn't work and doesn't make sense to put this onto your, in terms of financial toxicity. So working together as a community, together with patient advocacy groups, with stakeholders across the industry, but especially within the setting of precision oncology programs, it's important that if your doctor says, I don't think you're going to benefit from it, because the outcome of your test is in 99% of the cases negative. And if you spend this money out of the pocket yourself, you're going to be disappointed that this is a dialogue actually providing care and not limiting your options in the end. So this dialogue, I believe, is critical. And we at our institution, when we now hand out our informed consent forms, and we talk to our patients about starting next generation sequencing, we always say there is a chance that it doesn't work. There is a chance that it's non-informative. There is a chance that it's informative, but your insurance company is not going to pay for the drug, or there is not a clinical trial. So we start not by discouraging, but by being very open about potential outcomes. We manage expectations, and our patients, we've published on this, are okay with that, so they don't feel disheartened. And in the end, they said, well, we feel well taken care of because you explained it well to us, and the disappointment in the end was not so big, and they didn't feel that we were keeping anything from them. So this is a question very dear to my heart, and we need to really work together so that you guys are not just by proxy spokesperson of the pharmaceutical and especially the diagnostic industry.
Yeah,
I completely agree. Thank you very much.
I agree too, Siobhan. I have one more question, if that's okay, and I don't know. Oh, Andrew Green, do you want to go ahead, Andrew?
Oh, thanks, Dermot. Benedikt, that was a wonderful talk. I wanted to come back on Siobhan's point about, I'm a clinical geneticist, so I'm involved in cancer and non-cancer. And one of the difficulties for patients ascertaining genomics themselves is there are so many providers out there that you don't know what you're getting. Even if you try and find quality assured, externally assessed labs, it's very difficult, and they're all offering slightly different things. And many of them also don't necessarily have good clinical oncologists with expertise sitting beside the lab to provide the support, which then leaves the patient in a very vulnerable position, getting a large amount of genomic data, which is data, but it's not knowledge. and it's often very difficult to do. So it's something one would caution, both in the cancer genetics world, both for tumour and for germline, and also for germline in the non-cancer world. So it is tricky, and I mean, I can understand why people would do it, both in terms of trying to advance knowledge, but also for their own personal reasons. But it often could be a minefield.
Yeah, I couldn't agree more, Andrew. Thanks for this comment. And I just want to make one additional point. Even with very prestigious providers, often you get reports back and they list you a set of drugs on the very first page, and they list you 15, 20 clinical trials. We always joke about Korea. I don't know, this is a fixed term now, that we should send patients to Korea. I don't know why. And then secondly, again, and this is the same issue, if there are 5 or 6 drugs listed on the first page of a report, and you as a clinician say, well, but this doesn't work for you, I cannot give you this drug, it's not accessible, it's not working in your disease context, then patients feel as if they're cheated out of options. And this is where why we see a lot of these reports in our molecular tumor board. So we allow outside testing just to help our clinicians to put them into context, but then also give them a sheet of paper back that says, yes, we understand the molecular rationale here, but this is utter, sorry, my language, utter bullshit in a given clinical situation, you know. So, and this is also important to build these tightly knit communities.
Yeah. So Ben, this is one of the reasons we established the Irish Molecular Tumor Board, is because all this was coming in and we actually wanted a platform. So we accept outside and we just wanted experts to actually properly review the data, and then we feel it report. So this is exactly it, and it's a real issue. So I have Brian Bird has a comment.
No, I just wanted to ask Benedikt, has he any experience with tumor-informed minimal residual disease testing, and does he think it's ready for prime time? Because there's lots of people it would be great not to give adjuvant chemotherapy or maybe even adjuvant immunotherapy to. Und vielen Dank. Das war sehr interessant.
Ja, sehr gerne. That's so. Intellectually, it makes absolute sense. I believe the technology we've seen by Jeannie Tai and colleagues in her New England Journal paper, all of this makes absolute sense. Being very data prone is, I mean, why is no one running adjuvant clinical trials anymore? Because it takes 10 years to read them out. So I don't know if we're ever going to see clinical trials that will answer the question omitting chemotherapy based on minimal residual disease in a prospective fashion. So maybe we're going to see more and more real world data and prospective cohorts where it seems that these tools help. So if I'm meandering back and forth because I feel that those are powerful tools and I see a lot of potential here, but I fear that we're not getting the evidence that we would need to have to really sit down with patients and say, you do not have to have this type of adjuvant treatment. In breast cancer, I mean, looking at the breast cancer data on high, middle, and low risk patients, anti-hormonal plus minus chemo, those were 10,000 patients being randomized, and it took them a very, very long time. You can do this in breast with a humongous amount of money. I don't know if we're going to see it in a lot more indications in that way. So my answer is very powerful tool, would love to use it, would love to have more evidence and hoping for it.
Great. Thank you, Ben. Unfortunately, the clock has struck 5. So while I'd love to have, it was actually really interesting to see the Spanish clinical trial landscape and how do we have trials open in certain sites, but also have a degree of the trial delivered near the patient. I think is something that we need to work through as well in the future. But they're going. That's for another day's conversation. So thank you so much again for your time, your expertise, your insights, and your questions. And I would now like to close the session.
Yeah,
for those, sorry.
Just maybe from my side, thank you so much for giving me the opportunity to talk here today, and also for the very insightful questions. So my hand is extended if you want to do some intellectual sparring on the questions that we only touched upon. I'd be more than happy to get into informal, formal conversations so that we keep this dialogue going. And you have the slides, you can share them openly. And if anyone wants to get in touch on other questions, please do so. So my door is always open.
Thank you. That's much appreciated. It's good to have friends, and good to around the globe, and good to have experts to lean on. So thank you. Great. Claire had put up there, we've got a molecular tumor board next Tuesday. I think we've got cases. So, but for those of you who would like to join or be part of it, or submit a case for a future meeting, mtb@cancertrials.ie is the email, and Claire will coordinate it from there. And our hemato-oncology meeting then is the 20th of November. Thanks all.